So we just saw the basic kind of large scale anatomy of muscles. Now what I would like to do is focus on the muscle fibers themselves and describe some of the anatomy there. Okay, so I want to start by reminding you that muscle fibers are cells, right? So they're these specialized multinucleate cells. Um, so they have the regular cell parts. However, they are special cells. So we have some special names for parts of those cells, and then we have some specialized structures in them. So let's take a look at that. Okay, so first of all, starting over here, the plasma membrane of a muscle fiber has a special name. It's called the sarcolemma. Okay. Also, the cytoplasm of a muscle fiber is called the sarcoplasm. Um, so I guess that's not written here, but sarcoplasm, it's written, it's disguised. Okay. So those are, I think, kind of the main, like just names. And then we have these specialized structures, right? So we have this extensive endoplasmic reticulum inside of a muscle fiber, which we call the sarcoplasmic reticulum. And as you'll see, it plays a very important role in muscle contraction because it stores the calcium that is the signal for the muscles to contract, okay? Another thing that I want to point out is we have these T tubules here, okay? That's shown in yellow. Sorry, I guess I should have pointed out the sarcoplasmic reticulum is this blue structure that's shown in blue. T tubules are shown in yellow, and those are actually invaginations of the sarcolemma, invaginations of the plasma membrane. And they exist to allow action potentials to sort of penetrate to the inside of the muscle fiber. So you'll see that in the coming sections, okay? And then finally, I would say that these are the stars of the show. We've got thick and thin filaments, okay? Those are the actual contractile elements in the muscle fiber. All right, let's take a look. Let's take a look at those thick and thin filaments, okay? Oh, sorry about that. Okay, starting with thick filaments. Thick filaments are made up of this protein called myosin. A myosin molecule has sort of a globular head with a hinge region and a stiff tail. And the myosin molecules wrap around each other. So two of them will wrap around each other as illustrated in this diagram. And they will sort of line up kind of parallel to each other such that they are anchored to this kind of one anchor point. It's called the M line. So that's what a thick filament is. This is a thick filament, couple hundred myosin molecules in a thick filament. All right, let's talk now about thin filaments. The thin filaments are made up of a protein called actin. Actin is a globular protein. So they're depicted as these little spheres here. And so those actin molecules kind of line up to form a chain, okay? And then we have two actin chains that wrap around each other as illustrated in this figure. Also, I would like to point out to you that we do have a couple proteins that are around the thin filament, troponin and tropomyosin. So those are also proteins. They're like accessory proteins. And they also play an important role in muscle contraction, as you'll see. They kind of like regulate whether the muscle will contract or not, okay? All right, so zooming back out, we can see that the thick and thin filaments overlap as illustrated here. Okay, so we've got like sort of this interdigitation of the thick and the thin filaments, right? So thick filaments shown here in purple, thin filaments shown in pink, and you can see that there is this overlapping area, okay? When we are talking about muscle contraction, we think of these thick and thin filaments in functional units. Okay, so the functional unit that we talk about is the sarcomere. So a sarcomere is a unit as illustrated here. So if you go from this kind of zigzaggy pink structure called the Z line to the next zigzaggy pink structure, that is one sarcomere. Okay. It's a repeating unit. You have repeats of the sarcomere, right? So if you look to the right here, or if you were able to look to the right here, you would see a whole additional unit that looks like this. If you were to look to the left here, you would see a whole additional unit that looks like this. So we have these like long repeats of these sarcomeres, many, many, many sarcomeres, making up one, like the contractile structure within a muscle fiber. Okay. Oh, last thing that I want to mention on this slide is that this overlap is what's responsible for the striated appearance in striated muscle. Okay, so the dark, you can see that there are dark areas. The dark areas are where the thin and thick filaments are overlapping each other. And then the lighter areas are where you don't have overlap, right? So you can imagine like if you stepped maybe a couple 100 feet away from your screen, you wouldn't be able to see like all the details of this structure. You would just see a dark region here and a light region here and a dark region here and a light region here. And that's basically what we're looking at in this picture, okay? Okay, last thing for this video is that I want to say that the sarcomeres shorten during contraction. So basically what happens is that we change the amount of overlap between the thick and thin filaments. So looking at this figure, what you can see at the top here is what it looks like when the muscle is relaxed. When the muscle is relaxed, we have just a little bit of overlap between the thick and thin filaments, okay? When the muscle contracts, what happens is that the myosin pulls on the actin. So the myosin is the thick filaments. The myosin heads pull on the actin, okay? And they end up pulling the thin filaments closer to each other, I guess, so that you get more overlap between the thick and the thin filaments. As a result, on a macro level, what ends up happening is that this whole structure, this sarcomere, ends up shortening. Okay, so you can see that this distance is longer than this distance right here. Okay, and we call that the sliding filament theory of contraction. One more thing I want to mention, because I just realized I forgot to say it in the previous slide. So the myosin heads bind to the actin in the thin filaments, and we call that bond a myosin cross bridge. So it's like a kind of a non-covalent bond, but we have a bond happening between the purple little blob here, the myosin head, and this pink structure here, the thin filament.